Hello, and welcome to our comprehensive introduction to the world of cellular physiology. Today we'll be exploring the fundamental building blocks of life, eukaryotic cells. We will delve into their intricate anatomy and fascinating physiology, uncovering the secrets of how these tiny structures create the complexity we see in the world around us. Our journey today is divided into three main parts. First, we'll start with a basic introduction, defining what a cell is and understanding the fundamental differences between prokaryotic and eukaryotic cells. In the second section, we will take a detailed tour of the eukaryotic cell's structure, examining key organelles from the plasma membrane to the cytosol. Finally, in our third section, we'll explore the dynamic processes of general cellular physiology, including cell division, differentiation, and even programmed cell death. Let's begin. So what exactly is a cell? At its core, a cell is the basic, fundamental unit of independent life. It's the smallest entity that can replicate on its own. It's important to note that entities like viruses and prions are not considered independently alive because they require a host cell to reproduce. Now, life on Earth is categorized into two major types of cells, prokaryotic and eukaryotic. As we can see in this initial comparison, they differ significantly in size, complexity, and internal structure. Eukaryotic cells are generally larger, more complex, and possess a true nucleus, which gives them a high capacity for specialization. Let's look closer at these differences. The image here shows a prokaryotic cell which lacks a membrane-bound nucleus. Its genetic material is located in a region called the nucleoid. In contrast, the eukaryotic cell has a distinct true nucleus and a variety of other membrane-bound compartments called organelles. This structural difference has a massive functional impact. The key evolutionary advancement of eukaryotic cells is this internal organization, which allows for specialization and differentiation. This capability is what enables the formation of complex multicellular organisms like plants and animals. We now move to Section B, where we'll explore the structures within the eukaryotic cell, starting from the outside in. The plasma membrane acts as the cell's boundary, separating its internal contents from the outside environment. But it does much more than that. It maintains the cell's structural integrity, controls the transport of substances in and out, receives and transmits signals, and even enables movement. Its structure is a fluid mosaic composed primarily of phospholipids, cholesterol, and various proteins. This composition is also asymmetric, meaning the inner and outer layers of the membrane are different, which is crucial for its diverse functions. At the heart of the eukaryotic cell lies the nucleus, its control center. As the largest organelle in animal cells, it is encased in a double membrane called the nuclear envelope, which is dotted with nuclear pores to regulate traffic. Inside, the nucleoplasm contains chromatin, the cell's DNA wrapped around proteins. The nucleus has several essential functions. It protects the cell's genetic material and is the site of both DNA replication and transcription, the process of making RNA from a DNA template. While not all cells actively replicate, all cells perform transcription to produce the proteins they need to function. Next are the lysosomes, the cell's recycling and waste disposal system. These are small single-membrane vesicles filled with powerful digestive enzymes called acid hydrolases. To function properly, they maintain a highly acidic internal pH. Lysosomes are responsible for breaking down a wide range of macromolecules, from nucleic acids to proteins. They do this through processes like endocytosis, where they digest substances brought into the cell, phagocytosis for breaking down large particles, and autophagocytosis, which is the recycling of the cell's own old or damaged organelles. Vacuoles are membranous sacs that function primarily as storage compartments. Their importance varies greatly between cell types. In plant cells, a large central vacuole is crucial for maintaining structural support or turgor pressure, as well as for storing water and waste. In contrast, animal cells may have smaller, more transient vacuoles. A fascinating exception is the pulsatile vacuole found in some single-celled protozoans, which actively pumps excess water out of the cell to prevent it from bursting in freshwater environments. Peroxisomes are small but mighty organelles responsible for critical metabolic tasks. Their primary functions are oxidation and detoxification. They break down very long-chain fatty acids, releasing energy as heat. They also neutralize harmful compounds, playing a vital role in liver cells. A key process is the hydrogen peroxide cycle. Peroxisomal reactions generate hydrogen peroxide, a toxic byproduct. But the peroxisome also contains an enzyme called catalase, which safely breaks it down into water and oxygen. Defects in peroxisomal function can lead to serious genetic disorders like adrenoleukodystrophy, famously associated with Lorenz's oil. We've arrived at the powerhouse of the cell, the mitochondria. These complex organelles are the primary site of ATP synthesis, the molecule that provides energy for almost all cellular activities. They have a distinctive structure with two membranes. The inner membrane is folded into structures called cristae, which massively increases its surface area for energy production. Through a process called aerobic respiration, mitochondria break down sugars and fatty acids to generate ATP. It's also believed, according to the endosymbiont theory, that mitochondria were once free-living prokaryotic organisms that were engulfed by an ancestral eukaryotic cell, forming a symbiotic relationship. In plant cells and some protists, we find another remarkable organelle, the chloroplast. This is the site of photosynthesis, the process that converts light energy into chemical energy. Chloroplasts have a complex three-membrane structure, including thylakoid membranes stacked into grana. Photosynthesis occurs in two stages. The light-dependent reactions happen in the thylakoid membranes, where light is absorbed to create ATP and NADPH. These molecules then power the dark reactions, or Calvin cycle, in the stroma, where carbon dioxide is used to create sugar. Like mitochondria, chloroplasts are also thought to have originated via endosymbiosis. The endoplasmic reticulum, or ER, is a vast network of interconnected sacs that is the starting point of the secretory pathway. There are two distinct regions. The smooth ER is rich in lipids and is responsible for synthesizing lipids, detoxifying harmful substances, and storing calcium. The rough ER is studded with ribosomes, giving it a rough appearance. Its primary role is to synthesize proteins that are destined to be embedded in membranes or secreted from the cell. It also plays a role in protein modification and quality control. Once proteins and lipids are synthesized in the ER, they are sent to the Golgi apparatus, the cell's processing and distribution center. The Golgi is a series of flattened sacs or cisternae. Vesicles from the ER arrive at the CIST's face, travel through the medial section where they are modified, and are then sorted and shipped from the trans face. The Golgi packages these molecules into new vesicles and sends them to their final destinations, whether that's the lysosomes, the plasma membrane, or outside the cell entirely. All of these organelles are suspended in the cytosol, a dense gel-like fluid that fills the cell. Composed of about 70% water and 30% proteins and other molecules, the cytosol is the site of many metabolic reactions. It's also structured by an intricate network of protein filaments known as the cytoskeleton. This network, made of microfilaments, intermediate filaments, and microtubules, provides structural support, allows for cell movement, and acts as a highway for transporting materials within the cell. The cytosol also contains inclusion bodies like glycogen and triglycerides for energy storage. Now, let's shift to Section C and explore cellular physiology, beginning with the cell division and the cell cycle. This process, where a mother cell divides into two daughter cells, is a key characteristic of life. The process is highly regulated, especially in multicellular organisms, where division must occur for the organism's overall benefit. The cell cycle consists of two main stages: interphase, the longest phase, where the cell grows and replicates its DNA, and the mitotic phase, where the cell divides. This entire process is controlled by a series of checkpoints to ensure everything happens correctly and in the right order. One of the most incredible aspects of multicellular life is cell differentiation. Although nearly all cells in an organism share the same DNA, they can specialize to perform very specific functions. This process involves dramatic changes in a cell's shape, structure, and capabilities. For example, a red blood cell becomes a biconcave disc packed with hemoglobin to transport oxygen. A Purkinje neuron develops an extensive network of branches for complex electrical signaling. This diversity is achieved through the regulated expression of specific genes, turning different sets of instructions on or off in each cell type. Specialized cells don't work in isolation. They associate and communicate to build complex structures. This is the basis of multicellular life. Cells group together to form tissues. Tissues combine to form organs. Organs work together in organ systems, and finally, all the systems create a complete organism. This intricate organization is managed by highly complex mechanisms, including cell adhesion molecules that physically connect cells, cell signaling molecules that allow for communication, and gene expression that regulates these associations in space and time. Just as cell division is crucial for growth, cell death is essential for development and health. Many cells are programmed to die in a controlled process called apoptosis. This isn't a chaotic event, it's a regulated process that benefits the whole organism, such as by removing non-functional or potentially harmful cells. During apoptosis, the cell shrinks, its surface blebs, and it breaks into small, neat packages called apoptotic bodies. These are then cleaned up by immune cells called macrophages without causing inflammation. When this process goes wrong, it can contribute to a variety of diseases, including cancer, autoimmune disorders, and degenerative diseases. So let's quickly recap. We started with the basic definition of a cell and the key differences between prokaryotes and eukaryotes. We then toured the major structures of the eukaryotic cell, including the plasma membrane, nucleus, and various organelles, each with a specialized job. Finally, we explored key physiological processes like cell division, differentiation, and programmed cell death. The key takeaway is this: structure and function are intricately interconnected in eukaryotic cells. This elegant relationship is what enables the incredible complexity and versatility of life as we know it. Thank you for joining us on this tour of the eukaryotic cell. We hope you have a newfound appreciation for the bustling world inside every living thing.